Okay, in this video, this will be the first part of two parts for cellular respiration. And it is a process that happens both in plants and animals. And so I want to bring this up because some students think of chloroplasts as the mitochondria of plants, but that's not true. Plants have mitochondria, animals have mitochondria. They both use mitochondria for cellular respiration. Now, what is that? What cellular respiration is doing is taking reduced fuel sources, and we'll talk about what reduced means in just a minute, but carbohydrates, fats, proteins, these are all reduced fuel sources. They all have lots of carbons that are bound to lots of hydrogens, and they have lots of electrons that can be used to power a series of redox reactions that release energy that can be used to make ATP. And in so doing, those carbons become oxidized as CO2. Okay, and it's the oxygen that is sucking those electrons from these reduced fuel sources. And so that oxygen, after it sucks those electrons and hydrogens from the fuel sources, it becomes water. And those organic molecules, after they've had their electrons and hydrogens sucked away from them, they become CO2. All right, so glucose is one such reduced fuel source. And in fact, we're going to track the oxidation of glucose from the C6H12O6 here all the way to 6CO2. So we're going to look at all 6 of these carbons and see what the cell does to completely oxidize them into 6CO2. And what this does is it represents here our ATP cycle, in that we use cellular respiration to oxidize reduced fuel sources. That gives us the energy to recycle ATP from ADP and inorganic phosphate. And then that ATP is going to be used, because remember, it's so exergonic in the hydrolysis of that last phosphate group, it can be used to couple to all sorts of things and be used to do cellular work. And when it's used to do cellular work, it goes back to ADP and inorganic phosphate. And then cellular respiration's job is to put those two back together again to make ATP. Okay, so let's move on. The reduction oxidation reactions, or sometimes simply called redox reactions, are the reactions that drive cellular respiration. We are taking electrons from one molecule and transferring them to another and to another and to another, and these are all exergonic reactions that release energy, and that energy is being used to do work. What is that work? Making ATP. Now let's take a look at what it means to oxidize or reduce a carbon within an organic molecule. Here we have succinate, and we are turning it into fumarate, and this is an example of a redox reaction that occurs to organic molecules. And what you'll see here is that the reduced state of this molecule, so succinate, these two carbons are bound to 2 hydrogens each. So they are relatively reduced. The more hydrogens that a carbon is bound to, the more reduced it is. And now, this molecule, succinate, is being oxidized, meaning it's losing electrons and it's losing hydrogens. And so now you can see here at these 2 carbons, now they're only each bound to 1 hydrogen, right? So 2 hydrogens have been stolen along with their electrons. Okay, so where do those electrons go? Well, they go to these special electron carriers. In this case, they go to NAD+. That NAD+, when it grabs those 2 electrons, it becomes NADH. Now, enzymes that catalyze such reactions, where we are stripping of electrons from an organic molecule and giving them to NAD+, these are called dehydrogenases. So Take out that list of enzymes that you're keeping track of and add dehydrogenase to that list. All right, so also be able to look at two organic molecules and figure out which one is the more reduced and which one is the more oxidized. So if we take a look at glucose, the typical carbon in glucose looks like this. So each one of these vertices is a glucose. So that means this glucose is bound to one glucose. This carbon is bound to one carbon here, another carbon here, a hydrogen here, and a hydroxyl group here, an oxygen here. Now what you'll notice is this carbon, although it's reduced, although it's bound to a hydrogen there, it's not as reduced as it can be. It's also bound to an oxygen there. So glucose is definitely rich in energy, but there are reduced fuel sources that's even better than glucose. And I'll let you think about that, and we'll get back to that a little bit later. But right now, what we're looking at is taking that typical carbon that's bound to a hydrogen, another carbon, and an oxygen, and we are converting it here to a carbon double bonded to two oxygens. You cannot get more oxidized than that. And in so doing, those electrons that are passed on from our reduced fuel source here to the eventually oxygen, it's going to release energy. And that energy is used to make ATP. All right, so let's take a look at these electron carriers. This is nicotinamide adenine dinucleotide, and it looks like this. You will not be responsible for memorizing this structure. If you see it, however, you should be able to recognize it. And what we see is it's right over here at this N and right over here near this H. This is the active site. This is the site that's going to grab those electrons. And so here is our reduced fuel source from our food, the two hydrogens, and their accompanying electrons. Remember, we're used to thinking about hydrogens as protons, but don't forget that they also have electrons with them. So one of those protons and two electrons are going to go and be picked up by NAD+ to become NADH. And then one extra proton that's ripped from the food is just a byproduct. So it's this NADH that's carrying the two electrons that we just stripped from our reduced fuel source. Where does NADH take these electrons? Well, what it does is after it strips those electrons from our fuel source, oxidizing our fuel source eventually to CO2, our NADH goes to what's called the electron transport chain, and that starts off a series of redox reactions where we're passing those electrons from one molecule to another in a series, again, of redox reactions. Those reactions are exergonic. That energy is being used to do work to make ATP. The final acceptor of the electrons from our fuel source is oxygen, which when it accepts those electrons, it grabs a couple protons from solution, it becomes H2O. That is cellular respiration in a nutshell. But we're going to look at it a little bit closer. And what we'll see is we're looking at the oxidation of glucose. If we're looking at the metabolism of specifically of glucose, there are three important steps. First is glycolysis. The second is the citric acid cycle. And finally, we have oxidative phosphorylation. In this lecture, we're going to go through steps 1 and 2. And in the next one, we'll talk about oxidative phosphorylation in more detail. So let's take a look. First, glycolysis. We can get a lot of information simply by looking at this figure here. So let's take a look at what information we can get. Well, glycolysis is taking glucose and turning it into pyruvate. In fact, it turns it into 2-pyruvate. And notice where it occurs. It occurs in the cytosol of the cell, not within the matrix. And then with these 2-pyruvate that are made, they are actually shipped into the matrix where the second step occurs, citric acid cycle. We'll get to that later. Let's focus here on glycolysis. Now, I want you to know this. I want you to know what goes in, what comes out. One glucose goes in, 2 pyruvate goes out. I want you to know what electron carriers and ATP come out of it. Well, we get 2 NADH from it, and we get 2 ATP from it. The location, as I already mentioned, is in the cytosol, and this is the most primitive form of energy gathering metabolic pathways in organisms. Because as you will see a little bit later, as I'll make a bigger point about it later, it does not require oxygen, and I'll show you why that's the case again a little bit later. And it doesn't require any specific organelle. It happens within the cytosol, and it just requires a bunch of enzymes that are floating around within the cytosol. It doesn't need the mitochondria. Now, what I want you to do, and I'll show you how to do this, is I want you to track the energy. Right? There is an inherent amount of energy here in glucose. And as we manipulate glucose, as we oxidize glucose, I want you to be able to keep track of, well, where is that energy now? Where is that energy now? Where is that energy now? In addition, I want you to be able to track the carbons. Again, there are 6 carbons in glucose. After glycolysis, where are they? Well, pyruvate each has 3 carbons in them. 2 of them get made. That's 6 carbons there. So you're always tracking the carbons. Where are the carbons? Where are the carbons? Eventually, once we go through all of these steps, all of the carbons that started in glucose will eventually be in 6 carbon dioxides. Now here's glycolysis. There are 10 enzyme catalyzed steps. 7, 8, 9, 10. You don't have to memorize the enzymes, but for the purposes of appreciating the complexity of this, I want you to memorize that it's 10 enzyme catalyzed reactions. And what we end up with is, what we see here is that there's 2 steps in which ATP has to be spent. So one step here, one step here. So this first half of glycolysis is called the investment phase. But then in the second half of glycolysis, what we get is 4 ATP being made. That's called the payout phase. And so our net production of ATP in glycolysis is 2. Finally, we see here's a dehydrogenase. Here's our 2 NADHs that are being made. Remember, dehydrogenases are enzymes that catalyze the transfer of electrons from these fuel sources to NAD+ to make NADH. And then we go from glucose, a 6-carbon molecule, to 2 pyruvate. Each of them are 3 carbons, and so we haven't lost anything as CO2 yet. So where are the carbons from glucose now? In the 2 pyruvate. Where is the bulk of the energy now? Well, we have a couple, right? We've got a net of 2 ATP made, so there's some energy there. We've got 2 NADH there, got a little bit of energy there, but the bulk of the energy that's still left from glucose is right here in these 2 pyruvates. In fact, look at this 1 carbon here. When you think about the more reduced a molecule is, the more energy it can contribute. Well, look how reduced this carbon is. 1 C attached to 1, 2, 3 hydrogens, and then another carbon. There's a lot of energy still available to us there. And yet, look at this carbon here. Double bonded to 1 oxygen, double bonded to, or single bonded to another. That carbon's pretty darn close to being CO2 right there. And in fact, in the next step, this carbon is what we're going to lose as CO2. Now, one thing that I didn't really bring up here is that the bulk of ATP is going to be produced in the citric acid cycle. But there is 2 ATP that's produced here in glycolysis that's really important, a net of 2 ATP that's produced in glycolysis, and there's 2 ATP that's produced in the citric acid cycle. These ATP don't require the electron transport chain to be made, so how is it that they are being made? Well, remember, folks, and I talked about this for a reason, that there are some molecules, look at 1,3-bisphosphoglycerate, some molecules that are even more energy and higher energy than ADP, such that the kinase, remember kinases are enzymes that catalyze the transfer of phosphate groups from a molecule onto ADP or from ATP onto that molecule. Well, it turns out that 1,3-bisphosphoglycerate has higher energy than ATP, which means that this reaction going in this direction, phosphorylating ADP from 1,3-bisphosphoglycerate, is exergonic, and therefore kinase is catalyzing it in this direction, producing ATP. And there's another reaction like that here. Look at this. Phosphoenol pyruvate, it's phosphorylated, has more energy than ATP, which means that pyruvate kinase here is catalyzing the direction of the reaction in which that phosphate is going from the phosphoenol pyruvate to ADP to make ATP. So this is called substrate level phosphorylation, because the origin of that third phosphate group that we're putting on to ADP to make ATP is coming not from just an inorganic phosphate that's floating around solution, but from a substrate molecule. That's substrate level phosphorylation. And that is in contrast to oxidative phosphorylation that's happening in the mitochondria, in which that ADP, that inorganic phosphate for that third P, is just a floating around inorganic phosphate that's not attached to anything. So that's the difference between substrate level phosphorylation that's going on in glycolysis in the citric acid cycle and oxidative phosphorylation, what's going on in the mitochondria, in the electron transport chain. Okay, so now we have two pyruvates. Right? Each of them have 3 carbons. We need to further oxidize those carbons into CO2. And the yield that we get per pyruvate in this particular section is 1 acetyl-CoA, 1 CO2, and 1 ADH. But remember, glucose makes 2 pyruvate, and so if we're looking at the stoichiometry per glucose, it's going to be 2 acetyl-CoA, 2 CO2, 2 NADH. And this step occurs within the mitochondrial matrix. So the pyruvate that was made here in the cytosol is shipped into the matrix, and there is an enzyme complex called the pyruvate dehydrogenase complex. This is definitely another enzyme that you must put onto your enzyme list. And what this catalyzes, we should know it's a dehydrogenase, is that it strips electrons from pyruvate, giving them to NADH. There's our dehydrogenase aspect of it. But it's also a decarboxylation reaction, meaning we are losing this third carbon here as a CO2. So now all we're left with is these 2 carbons that had come from glucose. Now this CoA molecule, what's the purpose of this? Well, remember that when you phosphorylate molecules, one of the nice things about phosphorylating molecules is that it's a way of taking an endergonic reaction and turning it into an exergonic reaction. Well, the next reaction that this acetyl group has to be involved in is normally endergonic. But when we attach a CoA to this, we now activate this acetyl group, calling it acetyl-CoA, and now the next reaction it's involved in is in fact very exergonic. So this CoA is a way of activating this acetyl group. All right, what is that next reaction? What is it that acetyl-CoA is involved in? Well, it's involved in a set of reactions called the citric acid cycle. And the net yield per acetyl-CoA, so one round of the citric acid cycle, is 2 CO2. Ah, look at that. There's the oxidation of our final 2 carbons, turning this acetyl-CoA, 2 carbons, into 2 CO2, 2 carbons. 3 NADH, 1 FADH2, and 1 ATP. And don't forget, if we're talking about per glucose molecule, that generates 2 acetyl-CoA, and so you have to double all of those numbers. Now, the citric acid cycle occurs within the mitochondrial matrix, and it is a later evolutionary adaptation because it is an aerobic it requires oxygen. All right, now let me show you this here. This is what the citric acid cycle really looks like. It is a 8 enzyme catalyzed set of reactions, and it's a cycle and not a linear pathway because oxaloacetate is the first reactant and the final product. And this has some really important ramifications that we'll get into. But what's happening here, if we look at oh, by the way, I bring this up because you are not expected to memorize the structure of each of these intermediates or the names of the intermediates or the names of the various enzymes. But I do want you to have appreciation for the complexity, so I do want you to remember that it is 8 enzyme catalyzed steps where the final product is the same as the one of the initial reactants. So what has to go in? Well, 1 acetyl-CoA has to go in and 1 oxaloacetate goes in. And then what we do is we oxidize those carbons, boom, 2 CO2. In so doing, we have 3 NADH. We get 1 molecule of ATP from substrate level phosphorylation. We get a molecule of FADH2. This is another electron carrier very similar to NADH. And then we recycle our oxaloacetate. And so here then are the products. Now, the question is, what is peculiar about oxaloacetate? And this is the question I want you to ask yourself and to be ready to answer when we talk about it in lecture, which is, if I were to put 10 acetyl-CoAs into the citric acid cycle, what would be the net production of oxaloacetate? That is the question I want you to think about. All right, so let's take a look at where we're at. We have started with 6-carbon glucose molecule, and we have oxidized it to 6 CO2s. And in so doing, we have released a bunch of energy. What I want you to think about now is, where is the bulk of that energy that was once in glucose? Where is it now? In addition, I want you to tally this up. At this point, where we finished the citric acid cycle, all of those carbons are in CO2, how much ATP have we produced at this point? And from what pathways? How much CO2 have we produced and from what pathways? Of course, it's 6 because I told you that, but where do those CO2s emerge? Which pathways? How many NADHs have we produced and from what pathways? And finally, how many FADH2s have we produced and from what pathways? Now, the next lecture is going to focus right here. All of these NADHs and FADH2s, because remember, the whole point of this is stripping electrons from the reduced carbons, carrying them with our electron carriers, and using that in the electron transport chain to do a bunch of work to make ATP. So now we have to focus on where these guys go and where they dump their electrons to. All right, so the next lecture will focus on just that and the electron transport chain.